Thank you all for joining us on this inaugural webinar sponsored by the Algae Biomass Organization. We are so thrilled that so many of you are joining us and hope that you will come back for future webinars that we have on key topics regarding algae. I'm Mary Rosenthal, the Executive Director of the ABO, and I will be moderating a discussion today on commercial algae production and saltwater resource availability, which I think is a key topic for the industry to understand and explore. As many of you know, ABO is the trade organization for the algae industry. We are comprised of more than 200 member companies, research organizations, and individuals that are producing algae, providing materials or equipment, or using biofuels, fertilizers, and countless other products that can be sustainably produced from algae. In recent months, several companies have announced new demonstration facilities that will produce biofuels and other products from algae at a larger scale than ever before. With this expanding production comes questions about the impacts on natural resources. And water is one of the most important and most talked about. Today, we'll be hearing from 3 experts on the topic. First, we're going to hear from Dr. Stephen Mayfield from the San Diego Center for Algae Biotechnology on the ability of water to thrive in saltwater. Then we're going to hear from Drs. Mark Wigmosta and Erik Venteris at the Pacific Northwest National Laboratory on geographic availability of saltwater resources. Once these presentations are complete, we will have some time for questions from those listening in on the webinar. You can type your questions into your GoToWebinar control panel. During the Q&A period, I will select questions for our panelists to answer. I will do my best to host and answer all these questions, but in case we don't get to all of them, we will respond to your questions by email. So let me introduce our first speaker. Stephen Mayfield is director of the San Diego Center for Algae Biotechnology and the John Doves Isaac Chair of Natural Philosophy at UC San Diego. His research focuses on molecular genetics of green algae and on the recombinant production of therapeutic proteins and molecular biofuels using algae as a production platform. Recent studies from Steve's lab have shown the potential of engineering algae for the production of superior biofuels molecules as a source for renewable energy. Steve is also a scientific founder of Sapphire Energy, a company developing biofuel production by metabolic engineering of algae, and is currently the head of the scientific advisory boards of Sapphire Energy. So Steve, we're going to turn the controls over to you, and we're looking forward to your presentation.
Mary, thanks very much for the invitation to give the first webinar for the ABO. I'm happy to do that. I hope everyone can hear me now and see my screen. Unfortunately, I can't see you, so I can't see if heads are nodding or this is not getting through. I'm just going to assume it is until something shows up on my screen that says, where are you? So let me start with slide 1 right here. This is just an introduction for me. I'm Steve Mayfield. I am faculty at the University of California, San Diego, and I am director of the San Diego Center for Algae Biotechnology. That is a consortium of research scientists out here in San Diego. Includes academic guys from UC San Diego, Scripps Institution of Oceanography, San Diego State, SALT. But it also includes lots of commercial partners as well, Sapphire Energy, Synthetic Genomics, Life Technology. And what we attempt to do out here is to help take research discoveries from the lab and get those out into the commercial sector. So let's start right now with talking a little bit about saltwater resources and commercial algae production. So let's see, how do I advance my slide here? Okay, so what are the types of algae and aquatic biomass we think about? You know, I think most of us, when we say algae, we think of microalgae and cyanobacteria, which we kind of group together. There are also, you know, a number of people now who are starting to work on macroalgae, you know, the giant kelps. And I think what I want to say on this slide, and what I really want to point out, if you look at the little figure down on the bottom left, which is the phylogenetic tree of life, what you notice on there is that algae, you know, whether that's cyanobacteria, browns, greens, red, are really diverse organisms. The two little bottom lines, the little C. elegans and Homo sapiens, the animals at the very bottom, so that's worms and humans, and that's how close they are together compared to red algae and cyanobacteria. So these things are very far apart, right? But importantly, they all do one thing in common, and that's photosynthesis. And so when we think about these things, they are all photosynthetic organisms that live in water, take in CO2 and use sunlight to convert that into products. And there are a number of products that are actually sold right now from algae. A lot of people don't know this, or maybe some of you do and just aren't aware of how big those numbers are, but it's about an $8 billion industry worldwide. And what this slide really shows is that commercial production in algae today, the two biggest by weight are spirulina, which is a cyanobacteria, and chlorella, which is a green algae. And both of those are pretty big, 3,000 tons per acre per year of dry weight and 2,000 tons. One of those happens to be a saltwater strain, spirulina. Chlorella can either be freshwater or saltwater. Chlorella that's mainly produced commercially is a freshwater strain. And then there are a number of others that are produced now and sold. And you can see they're sort of equally divided between saltwater and freshwater. Haematococcus is one that many of you will know. That produces the pigment astaxanthine, which is sold commercially to color salmon. Very big markets there. And then, of course, dollar-wise, the biggest, I think, are the EPA, the omega-3 oil DHA, which is produced, sells for at $64 a gram, so $64,000 a kilogram. High dollar value, but not particularly large quantity. So if we look at and think about algae that we might use for commercial biofuel production, one of the first, I think, considerations that many people looked at was what is the oil content, and this slide simply is to show a few of the different algae out there and some of their accumulation of oil. And this slide is simply to point out that some of these are freshwater, some of these are saltwater. There's no correlation between oil content and whether you're grown in a freshwater or saltwater environment. And then another obviously very important consideration is biomass accumulation. Obviously, if we're ever really going to pull off biofuel production, we're going to have to get very high yields in terms of tons per acres per year. This happens to be shown in grams per liter. And I think what you can see is chlorella is sort of famous for having very high biomass accumulation. That's a freshwater species. Farther down is Nanochloropsis. That's a saltwater species. So again, productivity is not related to water type. Whether we grow these in freshwater and saltwater is a bit immaterial to how productive they are. That has to do with photosynthesis, and photosynthesis goes on quite well in either one of those environments. Clearly, one of the key considerations as we go to commercial production is going to be, can we do the genetic manipulations that will allow us to increase yield, to bring in crop protection, to get harvesting, and all of the other technologies, the efficiencies that we're going to need that really are going to drive the economics of this. And one of the things many of us think about is, what's our ability to manipulate genomes? And so this slide is simply to show what are the transformation technologies today for algal species out there. And again, the water types on the far right, you can see freshwater, saltwater, many different species, right, including diatoms and dinoflagellates and almost all the cyanobacteria. All of these things, we can do genetic transformations to them, meaning that we can introduce genes and get drug resistance. However, to really get this into commercial production, we're going to have some pretty robust technologies. Those robust technologies, the molecular techniques, have only been developed for a limited set of algae. Here's one of my favorite sets of pictures now. This just came out of my lab over the last couple months. We developed 6 different fluorescent proteins and put those into the green algae Chlamydomonas reinhardti. These are all nuclear expression. We target them to different parts of the cell. So as you can see, the little red cell up in the corner, that is the fluorescent protein M cherry targeted to the endoplasmic reticulum. The next one is cerulean targeted to the nucleus. We then cross those 2 strains together to get our M cherry ER cerulean nucleus, and then cross that with a strain in which we had put Venus, which is a green fluorescent protein, into the mitochondria. And when you cross all those things together, you can get that beautiful little guy down on the bottom, which is actually a live algae. That's a Chlamydomonas reinhardti, which is capable of swimming around with a blue nucleus, a red ER, and green mitochondria. And we now have one that also has a nice blue flagella. So those robust technologies are not available for all species yet, but we're working toward that. And so one of the things that we did over the last year was to take some of the strains of algae that we think could be commercial production in saltwater, and one of them happens to be Donnelliella tertialecta, and we asked, could the genetic tools that we developed for the freshwater algae Chlamydomonas be used? And it turned out you couldn't take the straight DNAs and just shoot them in. But what we could do is take the strategy that we used for Chlamydomonas and then go back and make the identical set of vectors, but in this case using the D tertialecta DNA. And this is simply one of the sets of vectors that we make to get expression of proteins in the chloroplast. We tried a variety of different industrial enzymes. Many of us think that co-products will help drive the economics of this. Clearly, one of the co-products that we can think about making are some of these high-value industrial enzymes. So we tried this set of xylanases, manases, phytases, and galactosidases. And then we transformed those into both Chlamydomonas reinhardti and D tertialecta. In Chlamydomonas, we use the Chlamydomonas vectors. Tertialecta, we use the tertialecta vectors. But essentially, the strategy was identical between these two. And this is simply a Western blot showing that we can get very nice accumulation of industrial enzymes in D tertialecta at about the same level that we've achieved in Chlamydomonas. Interestingly, it's sort of a different set of proteins. That's not all the same proteins accumulate in one algae or another. So there's some learnings we're going to have to go through to figure this out. But I think what this slide clearly shows is that we can develop robust genetic tools for saltwater species as long as we have a good understanding of the sort of genetics behind them. And so this last slide is just, where do we go from here? Well, I think with sequencing technologies the way they are today, so cheap and so quick, that as we identify new production strains, whether it's for us now Senodesmus, or for other groups it might be some of the other green algae, some of the chlorellas, I think as we identify those that have high productivity and high lipid yield, then it's actually only a very quick time until we can sequence those genomes, and then from that sequence information, develop all of the tools that we need to make these things productive. So I will end it there and pass this back over to Mary and wait for your questions.
Thank you, Steve. By the way, I would really love to use this photograph maybe on our stage or something of that sort when we do our Algae Biomass Summit in the end of September and October. So I'll have to talk to you about that. As a reminder,
ones that they actually show them swimming, because these things it's a little hard to tell on the screen, but they're actually alive. That's why the flagella, the blue flagella, are a little blurred there. Those guys are actually swimming off the camera as we were trying to take their picture.
I think that would be fabulous. As a reminder, you can submit your questions at any time in the chat window of your GoToWebinar control panel. I just want to make sure that everybody understands that, and I appreciate the multiple people that are here, which is fabulous. Now we're going to go into our next panelist. Dr. Mark Wigmosta is a chief scientist at the Pacific Northwest National Laboratory in Richland, Washington. His background is in environmental engineering and geology, and his recent research focus has been on the resources available in the United States for biofuel production. Mark and his team recently provided the first in-depth assessment of America's algal biofuels potential, giving available land and water, which is critically important as we continue to build this algae industry. Today, Mark is also joined by Dr. Erik Venteris. Erik is a research engineer at the Pacific Northwest National Laboratory. His background is in earth sciences with recent emphasis in subsurface geology and soil sciences. He is currently working on a national scale water and nutrient resource availability and cost modeling for the algae-based biofuels, with recent publications in Biomass and Bioenergy in Environmental Sciences and Technology. With that being said, I'm going to turn this over to Mark and Erik. The screen controls hopefully have been switched over to you, and you can start at any time.
Oh, great. Thanks, Mary. Just talking a little bit about the division of labor and the team. Mark been working on mainly on our water demand calculations and on our growth model. And my area is in the water resources, where I'm modeling water resource availability and cost. But what I want to talk about today is kind of show how those two work together to help us understand the overall algae cultivation system a little bit better. Excuse me. I have 2 points I really want to make in today's presentation. One, I want to introduce some of the national scale mapping we've been doing in regards to water quality and water availability. So we'll show that, and then I'm going to show you how our maps of salinity impact our choice of operating salinity. So there's an essential trade-off between the productivity of algae, or what they want to grow, what they want their cultivation conditions to be as far as salinity, versus what we want the salinity conditions to be to minimize our water resource demand and costs. So that's the two things we're going to talk about today. Oh, that doesn't work. There, okay. So we know that water availability is a key issue for algae, particularly in open ponds. Open ponds evaporate a lot of water, of course. NRC has put this as one of the sustainability issues of the highest priority in the recent report that came out. So in response to this, our biomass assessment tool that we've developed here, and what I'm going to be talking about today emphasizes open ponds, okay? I just want to clear that up. But we've developed capabilities to address this issue, or take this on head-on for the coterminous United States, okay? So, and we're starting to expand to look at worldwide issues as well. So we can predict algae productivity from a growth model that includes climatic and salinity data, and that salinity data is taken from collaboration with Michael Huseman, also at PNNL. And we have sophisticated models to calculate our water demand, and that evaporative water demand then drives our other main component of water demand, which is a blow-down stream that is required to maintain our pond salinity at a constant level. And that's going to end up being really important. And then where my area comes in is in the water source characterization itself. And we're starting to change our thinking and transitioning from the traditional fresh, brackish, saline, and try and get down to the essential categories that get down to the essential issues for algae biofuel. So we're proposing that we define a class called competitive freshwater, and this is freshwater water that has salinity low enough that agriculture and municipalities are wanting to use it. So the competition for this resource is acute. And then we have a second category, which we'd like to term non-competitive saline water. And here the salt content is generally too large to have a lot of people interested in using it. And that generally means too large for crops. But there are some industry people that may want may be able to use a little higher. But in general, not acute competition. And then the final group is the seawater, where we have unlimited supply. And a constant ion chemistry. So there's some real advantages of seawater. Of course, the big disadvantage is the cost of bringing it to a site. So you have to be near the coast, of course, to use it. Not quite as near, though, as some past literatures have suggested. At least that's our findings. So let's talk about national trends in freshwater availability and groundwater salinity. And then we're going to use those models and talk about what is the best operating salinity to choose for a given region that balances our production value and our water costs. So first I'll talk a little bit about some work that we did that is already been published. We have a model of competitive water availability for the U.S., and this was published in our recent article, Environmental Science and Technology. But what we did is we took the discharge for watersheds, and we calculated about a 25-year average. And then we made a very critical specification or assumption that we said the algae biofuel industry could have 5% of that discharge. Now, that's something that will probably not be cleared up until we have more clarity on the regulatory environment that's going to dictate how much of a watershed's given flow or groundwater that we can have as an industry. So this is what we're going with for right now. But you can see there's quite a contrast. Each one of these dots represents a site that has appropriate characteristics for an algae cultivation facility of 485 hectares or roughly 1,000 acres of pond area. And they're colored in gray where there is no fresh water available, and colored in black where we did have fresh water available. And you can see a marked contrast between the American Southwest and the American Southeast. Southeast has low evaporation rates, high precipitation, so a lot of our farm sites can be supplied with fresh water. In contrast, in the Southwest, we have high evaporation rates, low overall precipitation, which makes water resources in that area challenging and points to the need to better understand the potential of saline alternatives. One thing I would like to point out, however, is all water is salt water to a given degree. This is our current model. This is new. This is not published, showing the salinity of what we're terming the competitive waters. And you can see there are marked geographic contrasts in our average estimate of salinity. Obviously, geology is three-dimensional. We're boiling this down to 2 dimensions at this point as we do this national scale assessment with hopes of refining these things to a better, more resolved picture in the future. But what you can see in the southeastern United States is overall salinity levels of those waters we term competitive or fresh are lower than what we would find in the upper Great Plains or in the Arizona, parts of Texas, so on and so forth. So the impact there is that you're going to have more blowdown, and you're going to have more saline concentrate disposal costs in those areas where water salinity is higher. But every water has salinity, and every water would likely have a blowdown stream when we have an open pond situation. So also we're modeling the salinity in what we call non-competitive or brackish groundwater situation. This is the salinities ranging from 2,000 milligrams per liter up to 60,000 milligrams per liter. I will say that this model is far more uncertain than the previous one I showed you. We have an order of magnitude less data points, and the data is, this particular model is tuned towards Chlorella, so we don't model salinities above 60,000 milligrams per liter. So we have data that exceeds that. So for something that's a highly salt tolerant, we would have a different model. It's also important to note here, though, is that a lot of these, the high salinity areas are associated with oil and gas occurrence, sedimentary basins, and in this case, the spatial variations that we see are strongly related to geology. So it's more the occurrence of the sedimentary rocks and so on and so forth that dictate the location or the variability in the saline content of the subsurface waters in this case. So now we'll move on to introduce the national scale models. Now let's utilize those in some trade-off modeling, and we're trying to answer a basic question. You know, what is our bet on a regional basis, not on a site-specific basis, but more on a regional basis? What is our best choice of operating salinity? And finding that best choice of what salinity would we want to operate the pond at is really a compromise between growth rate and water costs. So in general, as that operating salinity goes up, and specifically as the distance between your resource salinity, the salinity of your water resource, and the salinity that you're operating at, as that increases, in general, our algae growth rate decreases and our attendant biofuel production. And we've looked at this for Chlorella and Enzymes. This graph here is for Chlorella. It's from Michael Huseman's data. And it shows a growth rate correction factor for a range of pond operating salinities. So Chlorella wants to have a fairly low salinity for optimal growth, and we have a linear decrease as that operating salinity increases, which means we have less overall oil or biofuel produced. However, our total makeup water also decreases as our salinity increases, and that means we may need smaller supply pipelines, need fewer supply wells, and then we have less saline concentrate that we have to dispose of. So overall, that works to decrease our costs. So then the question is, what's the optimal compromise? I don't have time to go over details. These are very complex GIS models, but the models are all tuned to connect each site up with its best, least cost source within a range of possibilities. And that's all we can really talk about. And these illustrate that for show the pipeline systems that are supplying a given site with water. So when we're done with that cost assessment, we calculate a biofuel production value. We subtract its water cost for each site, and we do that over a range of salinities. And then we take each site that has an economic parity where the value of the oil equals the water cost. It's highly unrealistic, of course, but it turns out that our choice of operating salinity isn't really sensitive to that choice in the analyses that I've done, but won't show you today. But anyway, that's the idea. So how do we maximize our total enterprise value? And that's what we're basing our costs on, or our choice of operating salinity on. So here's the operating salinity analysis for Salina. It's done on the Texas coast, based on 4,500 unit farm sites. And these thin little blue lines here are modeled pipelines supplying seawater to the site. And it actually does take into account local variations in seawater salinity along the coast, and that's mainly due to freshwater influx. So it accounts for a lot of things, the cost of constructing the pipeline, cost of operating it, so on and so forth. So then in our trade-off analysis, and this only applies to Salina, should always point that out, but we find that most of our material being brought in is at a salinity of about 35 grams per kilogram. And if we operate at 37 grams per kilogram, we have that value, but we seem to reach a maximum at about 40 grams per kilogram. So for that species operating on seawater, we want to maximize our total enterprise value, and that occurs at about 40 grams per kilogram, although you can see there's some flexibility there. If we had a goal where we wanted to reduce water consumption, we could choose a higher operating salinity. So moving on, now we're also going to do look at the operating salinity for Chlorella. And this analysis we're going to do for both competitive and non-competitive waters. And we have two contrasting study areas. We have 40,000 unit farms in the American Southwest, which we're going to compare to about 15,000 unit farms in the Southeast. And we'll do that for fresh and salt, saline or competitive or non-competitive waters. So looking at the results, this is in green. We have the total enterprise value, which is this water supply and concentrate disposal costs relative to biofuel value. So that's the green. We want to find the maximum on that green curve. But then I've also provided for you the water consumed per site. So as we go up in operating salinity, our total value of our enterprise increases as we have more economic sites and our water costs decrease, and that's offset a little bit by the decrease, of course, in the production of Chlorella. But also our water consumption decreases. So we can have many goals. We can either maximize our economic output or minimize our water output for our given choice in operating salinity. But what we find for the fresh or our competitive waters is for the southeastern U.S. By this analysis, optimal operating salinity is about 2 to 3 grams per kilogram. Now think about EPA suggested drinking water is about 0.5 grams per kilogram. So this is fairly salty water that we're operating our ponds in, even with a species that we generally consider as a freshwater species. And then when we go to the southwestern U.S., where our water supplies have more saline content and where our evaporation rates are higher, we find that an optimal operating salinity is more in the range of 5 to 7, or approximately double what we would find in the southeast. Our results are less certain for our deep saline waters or non-compete waters because of some of the limitations and difficulties in modeling these waters, but nevertheless we see similar trends. Decrease in the water, increase in the overall value of the enterprise, and in this case, for our waters that we don't expect acute competition for, we would expect a operating salinity somewhere around 15 grams per kilogram, and in the southeast somewhere between 7 and 10 grams per kilogram for those waters that we do not expect acute competition for. And then there's always a question, if you are using these non-compete resources, is chlorella your best choice? Well, that remains to be determined. So in our conclusions, I've shown you some regional trends in salinity, and I've shown you that when we, in general, when we look at the southeast versus southwest, those impact our water availability and our water costs, and also impact the way we operate our ponds. They can show you that, you know, in the southwest, we probably would want to operate at an elevated salinity relative to the southeast. It's also important to note, you know, this is obviously a very broad topic, and I've just given you some of the things that I'm thinking about currently, some of the latest results. We've got that one ES&T paper out that talks about in detail a trade-off analysis between fresh saline groundwater and seawater. So, you know, like I said, it's not a complete picture, but we still have some pretty serious issues, I think, that we need to address. What are the regulatory constraints that we expect on freshwater supplies and also on saline concentrate? You know, some states don't have a good regulatory environment for the injection of concentrates. You know, that would have to be developed to accommodate a new industry. And also, what are the temporal trends? What are the other competitors that are up and coming for our water resources? You know, there is always, of course, interest in desalinization of what I've termed non-compete. Well, in the future, that may be more competitive as municipalities look into desalinization to meet their water supply needs. And then, you know, the question of brackish and saline groundwater sustainability is really one of reservoir geology, and three-dimensional models at the national scale are not available. So that's a very difficult problem to address. And then, of course, there are other chemical aspects that are important. I've only touched on salinity, but we know that pH is important, the ion chemistry and the interactions between all those factors, so the interaction between salinity, pH, and ion chemistry. You know, and just an example, what is the growth performance? Some waters are sodium chloride is their ion content, but some others are bicarbonate. What would the difference in growth performance be? And what would the strategy be to deal with that? So there's a lot of questions that remain surrounding water, but I hope you can see that there are ways where we can kind of take the results of biology, combine them with an understanding of geology, and get a better understanding of the overall algae cultivation system. And that's it. Thank you.
I thought I was unmuted here. Okay, now I'm muted. Okay, actually, I am fielding the questions. And the first question is, first, how is saltwater defined as 3% as in the oceans? And does the same thing apply when we're talking about brackish groundwater with less than 3% salt concentrations? And that came from your presentation earlier.
So I think what the question is really asking there maybe is, when we look at all these different species and define them as either saltwater or brackish or freshwater, how are we defining those different waters? With the freshwater species, what we've identified is that they are not very sort of elastic in their tolerance for salt, meaning that some of the freshwater strains, Chlorella, Chlamydomonas, they're not very tolerant of going up in salt concentration. And converse, though, for the saltwater strains is that they seem to, most of them that we've looked at, seem to be pretty tolerant of having a decrease in salt. And then the other thing I want to point out, sort of, we've had this general discussion on saltwater, freshwater, and brackish water. Some algae have just an enormous elasticity in what they will tolerate. One of our favorite guys right now is Centodesmus, and that can go from very low, you know, the half-gram, gram levels of salt, all the way up to well above the 15 grams that Erik was just talking about. So there's some elasticity in this, and obviously that's evolved over time so that algae can survive under some different environments.
Thank you, Steve. Our second question is, how would you address the containment of genetically modified algae? Do you expect the same rule governing terrestrial crops to apply? How would microscopic nature of algae add complexity to the issue of containment? From a trade organization, I do have to, address the issue that, all of our companies are working within federal standards and within TSCA standards and things of that sort. But I would send this over to you, Steve, and I don't mean to put you on the spot, but you've had a lot of experience in genetically modified algae, and would love your comments.
Yeah, first I want to say that right now all of the strains that are outside, we have no GMOs outside in any open ponds anywhere in this country that I'm aware of. And I'm certainly not implying that we should put these things out anytime soon. This is clearly something that we're going to have to very carefully measure. And it's going to be regulated by the Environmental Protection Agency under the TSCA Act. They have a process in place now to evaluate these. We've actually been in discussions with them to start to look at some of the exact questions that you asked, which is, if we have a strain, if we have a genetically modified strain and we put it outside, how does that impact the environment around it? How does it survive? How is it invaded by other species? These are all important questions. Right now, what we know is that many algae, many different organisms in the environment tend to take up DNA from other organisms. There's been some great papers lately on what's called horizontal gene transfer. So I think this goes on all the time out in the real world. And so we're sort of in the process of measuring that. And I think that in the near future we'll have a much better, meaning this year, we'll have a much better handle on how that process is going to be. My expectation is that sometime in the near future, meaning in the next 2 to 3 to 4 years, some genetically modified algae will be allowed outdoors, but these will be modifications that are pretty benign. Certainly, I spend a fair amount of time, as you know, Mary, putting high-value proteins in, some of them even therapeutic proteins. None of those do I expect will ever be outside. But on the biofuels where we might be outside, I think, a gene that might modify lipid production, that would have some some utility in the biofuels arena. And I think that that's something that we can feel relatively confident that it will be benign out in the environment.
Thank you. The next question I have is, and I'd ask this to Mark and Erik, since evaporation in open ponds is a major sustainability concern, has PNNL evaluated the use of covered raceway ponds to reduce the rate of evaporation? Have you guys looked into that at all?
No, that's why issues are on our radar. But the key thing to remember is that the evaporation keeps our pond temperatures nearer to an optimum for growth. So temperature is the big trade-off there, and it might be covered raceways. I mentioned in the ES&T paper that my interest is in using evaporation retardant films that would allow some evaporation and some evaporative cooling. And I wonder if there's an avenue there where we could have an optimization between cooling and the amount of evaporation reduction. Certainly, I do think that's an interesting avenue, but we haven't modeled it specifically as such.
Well, and then I have another question for you, and this is, how do you monitor and optimize nutrient levels in water as far as higher salinity? I mean, how do you do that monitoring with all of the mapping that you're doing?
I think the easiest way to answer that question is we're not considering that. We're not delving into the complex geochemistry, biogeochemistry in the ponds. That's certainly an interesting issue, but that's beyond our scope right now. A lot of what we're doing is very simple resource demand calculations. I mean, at least simple is probably an understatement. If I showed the flow chart for what it took to make those graphs of operating salinity, you wouldn't want to see it. It's a complex model, but nevertheless, yeah, that's certainly interesting, but it's beyond our scope right now. So,
yeah, I think to clarify, right now, the growth model is driven primarily by solar radiation PAR and water temperature. We're kind of assuming that nutrient levels are optimal. We do calculate a nutrient demand, but the growth right now is driven more explicitly by pond water temperature and incoming solar radiation.
Well, we have many other questions that are coming in, and we will answer those by email. Unfortunately, as many of you know, we are a 501c6 nonprofit, so we only have so much time. And that being said, we appreciate everybody's attendance. And if you have additional questions, please feel free to send that either at info@algaebiomass.com or to myself at Mary Rosenthal at algaebiomass.com. We very much appreciate your time and support and attending this webinar, and we will have additional topics as we go through the course of the year. And we really appreciate all of your support with the algae industry, and I want to thank Steve and Mark and Erik for all of your time, and thank you so much. And we're going to be signing off on the webinar. Thank you so much.